other thing that we can notice about this is that there are enzymes or proteins involved in each of these processes. So none of this, although there are RNA molecules involved, none of this can happen without enzymes, without proteins. So that leads to a kind of chicken and egg problem, right? Where you have genes that store information, that encode proteins that make enzymes, and you need those enzymes to catalyze the reactions to copy the genes. So how do you get out of that problem? The RNA world is the way you get out of that problem.
The idea here is that at one point the chicken was the egg. Both the chicken and the egg were RNA. The RNA world describes a hypothetical era before proteins and DNA in which all the roles these play in modern organisms were believed to have been carried out by RNA molecules.
And Francis Crick the next year said that possibly the first enzyme was an RNA molecule with replicase property. Thus, a system based mainly on RNA is not impossible.
There are a couple of problems with this approach. More than 50 years passed since this idea came about. The understanding of molecular biology has undergone enormous progress, yet there is still no tangible evidence, example, or prototype to show that life without proteins is possible. 2. The earliest unambiguous traces of life already display the presence of proteins, so there is no fossil evidence for this proposed era. As far as we can tell, life and proteins appeared simultaneously on the planet. 3. When paradigm shifts occur in biology, the preceding worlds do not simply disappear without a trace. Prokaryotes did not disappear when eukaryotes emerged. Single-celled organisms did not disappear when multicellular life forms emerged, and asexually reproducing organisms did not go extinct when sexual reproduction emerged. There is value in simplicity, especially when it comes to natural selection. So why did the RNA world disappear completely? 4. There is selective pressure that prevents the accumulation of genetic material for low-complexity life forms. This is what makes the RNA world scenario literally impossible. There is a fundamental flaw in the reasoning of evolutionary biologists. They treat information, or more precisely data, as a free resource. The prevailing theory is that genetic material gradually accumulated by successive random gene and genome duplications. But storing, preserving, and copying data is not free. It's a costly task. An organism that spends its precious resources on copying unnecessary data is at a disadvantage compared to those which copy only the essentials. This pressure is proportional to the complexity and size of the organism. It hardly affects us, but this selective pressure is crushing for a tiny prokaryote. Howard Oakman at University of Texas demonstrated in a study titled Genomes on the Shrink that every clade of bacteria with genome sizes of less than 2 megabyte was derived from ancestors with substantially larger genomes. Subsequent studies confirmed that this evolutionary pressure exists both in vitro and in vivo, affecting free-living, parasitic, and symbiotic creatures, and it strongly affects both bacteria and archaea. As it turns out, the search for finding the simplest life forms has been going on for billions of years on an unfathomable scale, and modern bacteria is the result.
Possibly the first enzyme was an RNA molecule with replicase property.
In 2022, researchers at the University of Tokyo put this theory to the test by borrowing necessary enzymes from E. coli bacteria and acquiring the RNA template for the replication enzyme from a virus. They then began an evolutionary experiment. The original RNA template consisted of around 2,000 nucleotides. After only 200 cycles, the population was dominated by so-called parasitic sequences that were only 500 nucleotides long. This once again demonstrated the immense selective pressure for shorter nucleic acid sequences. The RNA genome shrank to a quarter of its original size in such a short time frame. This means that biological evolution could not have been started by a short piece of RNA, because the immense selective pressure to keep it short, or even shorter, would prevent the accumulation of genetic material. This force strictly prevents the buildup of longer sequences of nucleotides, demonstrating that the idea of a short piece of RNA leading to the origin of life is highly unlikely.